Hello students, myself Subhash Mishra. Today I am going to discuss with you the second topic of the biopractical. Students, before this I have discussed with you the syllabus as well as the first practical. Students, our first practical was related with pollen germination. In that I have explained you each and everything related with the pollen germination. And I think you have understood very well how we are going to prepare the slide for pollen germination in the lab. Now we are going to discuss the second topic that is also related with the pollen germination. In this practical, we are going to discuss the pollen germination, but that is not on the slide. We are going to discuss it on the stigma. So let us start. We will discuss the topic in different steps. So the first step is aim of the experiment. The aim of the experiment is to study pollen growth on the stigma of the flower. Now the second thing is material required. So for material required, we need fresh, mature, appropriate flower. After that, we need watch glass, petri dish, needle, brush, forceps, coloring pigment, saffronine or cotton blue, cover slip, plain glass, slide, and then microscope. These are the requirements for this experiment. After that, the very important thing is to have the knowledge about the experiment which we are going to do. So for this, we must know the basic things. I know you have studied lot in your theory portion. First of all, the pollen growth takes place when the pollination takes place. So let us see the basic structure of a flower. You can see that the lower part, it is the pedicel which is attached with the branch. Above that, receptacle is there on which the four whorls of the flower are arranged. Then you can see the leaf-like portion, that is sepal. Then the attractive part of the flower is petals. Petals are the attractive parts. They attract the insects to sit on the flower and do the process of pollination. The next part is stamen. Stamen contains anther and filament. The next part is pistil or carpel, which contains stigma, style, and ovary. In ovary, there are presence of ovules. So this is the basic structure of a flower. So students, now you have the idea of the basic structure of a flower. When the pollen grains, they fall on the stigma. After that, the process of pollen tube growth starts. Students, in my previous video, I have explained you the structure of pollen grain. So I think you know very well that a pollen grain is double layered. Outer layer is XII and the inner layer is intine. After that is possess two types of cells. The one is the vegetative cell and this one is the germ cell. Vegetative cells are also known as tube cells, and these germ cells, they give rise to two male gametes or the sperm cells. Now you are well known about the pollen grain, its structure and the basic structure of a flower. Students, now when the pollen grains they fall on the stigma region, when pollen grains they fall on this stigma region, then the stigma region it contains sugary fluid. This sugary fluid it is absorbed by the pollen grains, and pollen grains when they absorb this sugary fluid, then you know very well that the inner wall of the pollen grain is thin. So when it absorbs lot of sugary fluid, then it get ruptures. The rupturing portion is micropore. So from micropore, pollen grains they start arising a pollen tube. So the pollen grains which are here on the stigma, so one of the pollen grain start arising its pollen tube. This pollen tube they arise through the style and moves towards the ovary to ovule region. Pollen tubes they contain two male gametes or sperm cells. This pollen tube when it enters through microcoil, then it ruptures this ovule region. This ovule region it contains from microcoil end it contains one XII, two synergids, and in the center two nuclei cells, and at the top three antipodal cells. Now when this tube enters in the ovule region through microcoil, then it ruptures one of the synergid cell it through its cytoplasm along with the two male gametes in it. One of the male gamete it fuses with the egg and give rise to the formation of zygote, which later on becomes the embryo. This is the first fertilization. Now the second male gamete it fuses with the two nuclei, and this process is known as triple fusion. And since here again the male gamete is fusing with the two nuclei, then this is the second fertilization. This is second fertilization. So after this second fertilization, then the formation of endosperm takes place, which later on give rise the nourishment to the zygote for the formation of embryo. So students, this is all about the double fertilization. So in this way the basic concept which was necessary for you that I have given to you, and I think you have already studied lot about it in the theory classes also.